Great. So thank you, Meghan Azad, for agreeing to chat today. It's great to have you talking all about breast milk and the infant microbiome, as well as breast milk microbiomes. So maybe we can just start off by you giving us a little insight into who you are and how you got into microbiome research and what it is that you work on today.
Sure. Well, thanks for having me. This is my favorite thing to talk about, so it's a pleasure. So I am a professor at the University of Manitoba in the Department of Pediatrics and Child Health. I'm also a research scientist at the Children's Hospital Research Institute of Manitoba. And I co-founded and direct the Manitoba Interdisciplinary Lactation Center, or Milk with a C for short. And I lead a bunch of projects related strongly to infant nutrition and microbiome development. So I have a Canada Research Chair in early nutrition and the developmental origins of health and disease. And sort of what brought me to here, I can't believe how time flies, but I just realized it's been 10 years since I started my lab. So we're going to do a little bit of a 10-year party in a couple months. But when I started my lab, it was with the intention of understanding how does breastfeeding shape the microbiome. And the reason I was interested in that is that I did my postdoc with Anita Kazurski at the University of Alberta. And my project there was about the infant microbiome at a time when, thinking back a decade, and even more than that, this was a pretty new area of science. So in Canada, there had just been a call from the government to submit applications for team grants in microbiome research. And so Anita, my advisor, had put in one of those grants and been awarded it. And the premise of her grant was to understand how the infant microbiome plays a role in the development or prevention of allergies and asthma in children. And it was a bit of a piggyback onto a study that was just getting started in Canada called the Child Cohort, the Canadian Healthy Infant Longitudinal Development Study, or CHILD. And that study was formulated with the idea of understanding why so many more kids in today's generation have allergies and asthma compared to even a few generations ago, with the recognition that genetics are important, but they obviously aren't the whole picture because our genetics haven't changed in a couple of generations. So that study was formed to take a really big look at genetics and the environment as the thing that has been changing to understand why are there so many more allergies today. And the microbiome actually wasn't part of the design of that study from the outset because it was designed back in like 2005, 2006 at the planning stage. So it was really before microbiome came on the scene in a big way. But luckily someone in designing that study had the foresight to say, let's collect stool samples, diapers, amongst all the other many samples we're collecting: blood, urine, house dust. And so a few years later, the study's getting started. They're recruiting, it's a birth cohort, so recruiting pregnant moms. And at that time is when the Canadian government put out this call for microbiome research. So Anita thought, hey, I'm part of this big child cohort study. We have diapers, we could analyze the microbiome. Let me put in a grant. And so it was awarded. And right at the time I was starting to look for a postdoc, she was looking for a postdoc to work on this project. So it was really good timing. I had not done my PhD in anything related to the microbiome or child health for that matter. I had done it on cell signaling and cancer, but I was sort of not feeling that topic area anymore and looking for a change. And this appeared to be a neat way of combining my molecular science background with a new type of research question more applied to population health. So I worked for 3 years with Anita, learned all about the microbiome. The study was still just getting going. So fun fact is that it's still going today. So those babies are now teenagers and we're still following them. But at the time, many were still being born. So we didn't have the ability yet to say, how is the microbiome influencing allergies? Because we still had to wait for them to grow up and get allergies. But we said, well, let's just take a first look at the microbiome. What does it look like? And what's shaping it? And so my very first paper in this field was with Anita. We looked at 24 babies of this study that now has over 3,500 people. So it was pretty small, what we considered a pilot analysis. And we just profiled the microbiome using 16S. And we saw that the main things shaping the microbiome of these 3 to 4 month old babies was how they were born, so vaginally or by C-section, and how they were fed, so breast milk or formula. And that was an exciting experience to be part of that research. And it was, featured on the cover of the journal and in the newspaper, and a lot of people were excited about it, which was new to me because the research I had been doing prior to that, very important, but very technical, self-signaling, kind of very far from application. So it was fun to me to see that, wow, like the average person is interested in babies and their microbes. And so I really, I guess, caught the bug no pun intended, and thought this is something that I want to pursue. I want to understand at a deeper level, like why is it that breastfeeding is so important to the microbiome? And also kind of strategically, I guess, I knew that the cohort had also collected breast milk samples. And so we had the opportunity to not just look in more detail at how the babies were being fed, because I looked at just breastfeeding, yes or no, but we actually had a lot deeper level questions than that, which I've gone on to explore. But also we had the breast milk itself, so we could start looking at what's in the breast milk and how are those different components related to the gut microbiome.
Right. Yeah. And now you're kind of known as the breast milk microbiome person around the world. And you've described that so nicely now that we know, partly from your research, but other people's research around the world, that the breastfeeding is probably the strongest determinant of what an infant's microbiome looks like. Um, maybe if I can, we can discuss a little bit of historically and evolutionarily thinking about breastfeeding. Mammals breastfeed, and it's a perfect source of nutrition for us mammals. Can you talk us a little bit about kind of maybe what you know about breastfeeding in the natural world and breastfeeding, historically in humans, and how our approaches to breastfeeding have changed or are changing?
Yeah, so I think, the average person might not realize, although of course it makes a lot of sense once you think about it. Mammals all have mammary glands, that's what they're named for, and therefore they all feed milk. They all lactate, make milk, and feed milk to their young. That's like the defining characteristic of mammals. And so there are many species, humans included, that rely on lactation. We don't say breastfeeding for other animals that don't have breasts necessarily, but this way of feeding our babies. And so every one of us humans alive today really owes our existence to breastfeeding, even if we ourselves weren't breastfed as infants, because in modern history that has changed. But that's obviously then a hugely important physiological system of our species and all mammals. So sometimes I think it's a little wild that we don't know more about it, considering how important it is. And I think a good way of putting that in perspective is in medicine, you have a specialty for everything. You have, nephrologists that specialize in kidneys. You have people that specialize in brains and kidneys and livers and every part of the body, but there's no specialty for lactation. Until actually last year. There finally is one now, at least in the US and Canada. So it's kind of crazy that it's taken this long. And I think there are many kind of historical reasons for that. One of them being that it's the so-called women's health issue, although I'd argue every baby, regardless of their sex or gender, needs food. So I wouldn't put it in a women's category, but that maybe has been the case historically. So I feel like in a way I'm lucky to be here in the right place at the right time, because talking to my senior colleagues who've been in the field of breast milk and breastfeeding for decades, there's a real sense of frustration. Why is nobody understanding that this is important? There's not enough funding for this research. There's not enough people doing it. And that is changing, and I'm encouraged to see that. I think we have more progress to make, but there have been, for example, directed calls for research on women's health broadly, and then lactation specifically from the US National Institutes of Health and others in the last 5-10 years, which is great to see. But yeah, it's hugely important. Our survival as a species depends on it. That's true for all mammals. And there are commonalities then in lactation across all mammals. And for people who are from the field of genomics, they'll do things like look across all mammals and see what are the genes involved in lactation, how are they conserved, or how are they different, can that tell us? But at a level of the milk composition itself, I think it's really neat that we all make milk, all the species, but the milk is very different and kind of optimized for each species, which again makes sense. So a baby cow, for example, and a baby human are very different. A cow is going to get up and be wobbling around, and it needs really strong muscles really quickly. So cow milk is going to be optimized for that. Whereas a baby human is not really using much of its muscles to walk around for many months. Instead, it's devoting a lot of its energy to brain development. And so human milk is optimized for that. And then it's not just this species specificity, but also the dynamics of milk. So a baby cow or a baby human is growing really fast, and its needs are changing as it develops over that period that it's consuming milk from its mother. So the milk is changing over time as well, which is something that we're studying. And even we think of day by day, month by month, but even within one day, from the morning to the night, we're learning that milk has differences, and that can contribute to infants developing their circadian rhythms. And then there's a lot of individuality to it too. So it's not that every cow is making the same milk or every human is making the same milk. It depends on that mother's genetics and on her environment. So, a mother living in Canada or the US or the UK, a high income country, is going to produce different milk from a mother living in a really hot country or a country with other types of pathogens that are sort of endemic in the environment. And so that's something to keep in mind as well. And so by learning about sort of the differences and the changes in human milk, I think is really important. And we also have a lot to learn from other species and researchers that have been working in that area, which, to be honest, you could argue there's more known about like cow milk than human milk, because
Yeah, yeah,
cow milk
yeah.
is dairy milk, right? Like there's money in that. So there's been a lot of research going on. So
And I mean,
yeah,
I guess one of the main reasons that we have neglected this is because of changing human behavior and the fact that we are, in the natural world, one of these only, well, probably the only mammal or the only species that can lactate, produce milk, and voluntarily, you know, decide not to for whatever reason that may
yes,
be, and shorten that. And so you mentioned nicely how our genetics take generations to change. However, you could argue that over the last 100 years, our environment has changed drastically. Uh, no more so maybe than in the first few months of life. The exposures that children are getting in the first months of life are very different to what they would have, generations or hundreds, if not
yeah.
thousands of years before. So maybe can you just touch a little bit on changing practices towards breastfeeding in humans with industrialization?
Yeah. So of course, until maybe 100 or 200 years ago, every baby got breast milk. That's what happened. But then with industrialization and technology, there started to be, well, could we have something a little more convenient? Could we develop a scientific based formula to feed babies so that mothers aren't having to breastfeed? And also maybe we can develop something even better because we have technology and knowledge and science. And so infant formulas were developed, and they are based on cow milk, which is another thing that I think the average person doesn't necessarily stop to recognize. But formulas are based on cow milk. They're highly processed versions of that. So there's a lot of processing and supplements and that goes on with making formula from cow's milk, with the intention of making it closer and closer to human milk. So better for human babies. And that involves, adding certain components, making sure that certain essential components, certain vitamins, for example, are there at the recommended level. And so it's continuously improving, but certainly the first versions and even the current versions are not human milk. And from what I just said about how human milk changes over time, even from morning to night, and it's different for different moms, like that's something we really can't, even with the most possibly advanced technologies and knowledge, replicate. But so these formulas came on the market, and if you look sort of historically at trends in breastfeeding, you see sort of everyone's breastfed and then a dip because now we have these alternatives. And then an uptick. And so there was this, okay, we have an alternative, no need to breastfeed. And then it took a couple of decades for kind of research to catch up and people to start thinking, well, we're assuming this is better, but is it really? And like collecting the data. One of my sort of epidemiology heroes is Dr. Cesar Victoria. He's an epidemiologist from Brazil. And he's been recognized with some really large international awards for his work on breastfeeding. And he's done lots of other things too, but he was one of the first people around the early 80s to start collecting data and showing that actually formula-fed babies in his home country were at higher risk of things like diarrheal disease and infections and even mortality. And so to first of all notice that and say, whoa, and then why is that? Is there something in human milk that we missed that we need to be adding? And even took it a step beyond that to take it to his government and policymakers in Brazil and say, look, I have this evidence that breastfeeding is ideal for babies. Like, what are we going to do to promote this and turn this shift around? Because people are assuming that formula is better or at least sufficient. So he's done a lot of great work and was inspirational to me, I would say, when he won that award. The first one was a Gardner Award in 2017. So I had just started my lab. And we can talk a little bit about, there's a lot of emotion and controversy in the field of breastfeeding. And as a new PI, I was thinking like, is this where I want to focus all my efforts? Because it's difficult. And it was also before these new grants in the area. And I thought, is this going to be like too challenging to pit myself in as a field? But seeing that recognition was like, okay, people are starting to catch on. This is important. But anyways, yes, so there was definitely a downtrend in breastfeeding, and it's now in many countries, I'd say Canada and the UK included, on an uptrend, but we have a lot of ground to make up because of this evidence coming out and showing, yeah, there's a lot of important things in mother's own milk that we can't replicate in a formula and that are important at least for a certain amount of time to give to your baby. And so the public health messaging and things that go with that are in progress. But
Yeah.
yeah, there's definitely been some historical changes and trends over the last 50 years, 100 years.
Yeah. And still, large geographical differences around the world,
Hmm.
as you mentioned, in non-industrialized countries, it's a free source of food and nutrition for babies. And so the babies tend to be breastfed for much longer there, and tend to be fed exclusively rather than maybe mixed feeding.
Yeah.
So, I mean, I suppose it's important to state that we know the WHO recommendations are for exclusive breastfeeding, and babies don't technically need anything else other than breast milk for the first 6 months of life, and
Yeah.
then they're weaned onto solid foods. But still, the recommendation is to continue breastfeeding up until at least a year, maybe even 2 years, and
And
beyond
beyond. Yeah.
and
If
beyond
it's,
if wanted. Yeah. Um.
Yeah, I mean, and
So
that's
what?
different in different countries too, in terms of
Yeah.
the practices, right? And
Right.
it's interesting that in high income countries today, the trend tends to be mothers with more education, maybe more opportunity, are more likely to breastfeed longer,
Mm,
possibly because they've received the education and knowledge that this is really beneficial, possibly because they have more opportunity to take time off work when they have a baby, and therefore be at home and be breastfeeding. And it tends to be the lower socioeconomic status segment of the population that has lower breastfeeding rates. But if you look in the past, or in other countries today, that can be reversed. So in some countries, it's sort of like, well, being able to afford formula is a status symbol. So people associate feeding babies formula with sort of better wealth. And so you might see the opposite trends. Or similarly in countries like ours, several generations ago, it was again, well, working moms are busy and they can afford it and they'll feed formula. So it was the opposite to what we see now.
mm. So before we stray too much, I suppose, into the social science behind breastfeeding,
Yeah.
and maybe we'll come back to the science of it all. So you touched on that kind of historical research where people were kind of looking at what components were in breast milk, and realizing that maybe these replacements that we had in formula didn't have all the ingredients that were there. So we know, maybe we can run through. We know that there's like sugars and fats and basic things like that in breast milk, in human milk, that are similar to what you maybe could get in cow's milk. But there's also plenty of other things there which we are only maybe beginning to replicate, or in some cases haven't been able to replicate. So maybe we can start off talking about human milk oligosaccharides, because anyone in this field will be aware of
Yeah.
them. Maybe you can tell us what these are, what are HMOs, and why are they such an integral component of breast milk when we're thinking about a baby's microbiome?
Sure. So yes, as you mentioned, there are components in milk, I'd say the nutrients that we're going to find in formula. And so these are fats, lipids, sugars, mainly lactose as the one that the baby will digest, and then micronutrients, vitamins and minerals. But then I think of everything else on top of that as sort of the non-nutritive bioactives. So things that the baby's not digesting for food or energy, but that are having an important influence on, the baby's immune system, for example, or brain development or other things besides just metabolism and calories. So HMOs are one of those. And actually HMOs are the third most abundant solid component of milk. So if we sucked out all the water and then measured what's left, you would first have fats and lactose. But then even before proteins, which you'd think, well, protein's an important nutrient, you actually have more HMOs than protein in human milk. And that's one thing that's different in other species of milk, perhaps. But in human milk, we have more HMOs than protein. So these are human milk oligosaccharides. They are complex carbohydrates. So they are not digested by the baby. And so, for a while it was maybe, well, these things must be important. The mother is devoting a lot of energy to produce them and put them in the milk. But what are they doing there if the baby is not digesting them? And with the surgence of interest in the microbiome, it's become very clear that, oh, they're there to feed the baby's microbes. Because the microbes in the gut can digest them, at least certain microbes, which is another interesting point. It's not every microbe that can digest these HMOs, but
Mm,
certain ones that turn out to be beneficial for infant health. And so certainly, I don't want to discredit the early researchers of HMOs. I think since HMOs have been known, it's been known that certain bacteria can eat them, but it's really in the last 10, 15 years that people are paying so much attention to the microbiome that it's like, oh, that's what these are there for. Let's understand the HMOs.
mm.
And so I got interested in them for that same reason, because I was interested in the baby's gut microbiome. I was at a microbiome conference where my now collaborator, Lars Bodie, was giving a talk about HMOs, which he was studying in rat models and also in some human samples. And that was my first real introduction to the fact that, first of all, these things exist in human milk and they're highly abundant. And essentially, we don't know a lot about them, what they do, like how variable are they, what drives that variation. How are they important to infant health? And so I immediately went up to Lars after his talk and said, you have this technology to measure these important things. I have a cohort of babies where we've already measured their gut microbiome, and I have lots of milk samples. Like, we should work together. So we did, and we now work on many projects together, profiling HMOs. And one of the fascinating things about them is in human milk, we still don't have the official number, but there are arguably 100, if not 200 different HMOs produced by women. So these are complex sugars in slightly different physical formations. So they're all HMOs, but they're structurally different. And that structure can impact their function, of course. So they can have different functions based on their structure. And some of them are, present at high amounts, some of them at low amounts, and we're still learning about that. But in other species, for example, it's estimated that cows make about 40 different, well, we wouldn't call them HMOs, BMOs, bovine milk oligosaccharides. And mice, for example, which of course are also mammals, they only make 2 oligosaccharides in their milk. So this seems to be one of the things that, as I guess arguably more complex or higher evolved species developed, they also increase the complexity of their human milk oligosaccharide profile. And so it's one of these components of the milk that is very species specific. So maybe it's one of the things then that helps milk be optimized for different species. So that for me is a fascinating reason to study it. And we think a lot about what they do for the gut microbiome, because we do know that certain microbes in the baby can consume them. But I also have to acknowledge that's not their only function. So we know they're consumed by the gut microbes, but they also, we are learning, have other functions. So they can interact, for example, directly with the infant's gut epithelium. They can also be absorbed in small amounts into the baby's circulation, which allows them access to then other organs of the body. And we even know now that they are found in a mother's amniotic fluid and in her blood when she's pregnant, towards the end of pregnancy. So maybe the word human milk oligosaccharide is like a misnomer in a way, because it's not only in milk, although that's certainly where we found it first and we study it most.
Wow. So babies are almost getting breast milk before they're born, they're getting some of these components.
Exactly.
I didn't know
Yeah,
that.
they're
That's
ingesting
fascinating.
them through the
Wow.
amniotic fluid, which makes me think like maybe that is kind of priming their gut before they even reach the world and have microbes present to like receive these HMOs.
No way. And they're
Yeah.
produced in the mammary gland and being passed through the placenta. Wow.
Yeah, so they're like, and so this, which makes sense why they're found in the mother's blood as well, because they have to
Yeah.
get from the mammary gland like
Mm.
through the placenta, and so they're in the blood to a certain degree, and sort of still learning. There's someone I think comes from Lars's lab years back who's been studying them in circulation and amniotic fluid and showing, they're different maybe in mothers with different metabolic conditions.
Wow.
Yeah, so looking at what do they look like before the baby's even born, and then what do they look like in the milk. With Lars, one of the things we chatted about after that first encounter was, in the child cohort where we have a pretty large sample size, we know a lot about the moms, like, what is their diet? What is their lifestyle? What is their ethnicity? We have their genetics. So could we use that information to understand more about what drives the huge inter-individual variability in HMO profiles? Because it's highly like individualized in different people. So we've been working, for example, to link the genetic data we have with the HMO data to understand which genes are driving the variation in HMO profiles.
Okay. And I mean, what is the headline of that? We know that what the main environmental influences are on the gut microbiome. We know there's a small genetic effect. There isn't a very strong genetic effect on the gut microbiome. I mean, what are the main influences on HMO composition? Is it genetic
HMOs.
or is it
Yeah,
environmental?
genetic is a big one. So almost
Really?
maybe a little bit the reverse of what you just described for the
Okay.
microbiome, where yeah, there's a
Okay.
small genetic effect, but it's highly environmental. For
Mm.
HMOs, we see the opposite, where it's highly genetic and there's a
Right.
bit of environmental influence as well.
Interesting.
But for example, one gene that we know a lot about is called FUT2 or fucosyl transferase 2. So this is an enzyme that contributes to glycosylation pathways, not only for HMOs, but for like all the glycans in your body. And it's a gene that has a common SNP, so a single nucleotide polymorphism, 1 letter difference in your DNA. And based on that SNP, you either have this enzyme that's functional or you don't. It's truncated, it doesn't work. So it's kind of like one of those rare traits that's either on or off. Like you are, it's called a secretor if you have it, a non-secretor if you don't. And you can see that like black and white in a milk profile or other types of tests where either you make certain HMOs or you do not make them at all. And I mention this because one of the HMOs affected by this gene is called 2-fucosyl-lactose or 2FL. And if you're a secretor, it's the most abundant HMO that you make. It's there in huge quantities. If you don't, then you don't make it at all. You make other HMOs in different quantities to almost like make up for it. So it's a very big signal. And this is also not like a rare mutation. About 25% of people are non-secretors. And so speaking, we talked a little bit earlier about evolution. Evolutionarily speaking, you don't find 25% of people with a like mutation or DNA difference if it's not sort of good for
It's not
something.
good. Okay.
Right. And
So we
so
don't
it's sort of
know
like,
from
well,
historical evidence that people who are non-secretors are, less fit. There's no like fitness
so we
effect
do
to.
know some things like, so for
Okay.
example, non-secretors are completely resistant to norovirus infection because the virus
Wow.
uses one of these glycans sort of controlled by that gene and enzyme to infect host cells.
Right.
So this is unrelated to milk, but so that's an advantage that non-secretors have. So that could
They
be one reason
cannot
why.
get infected at all. So this is winter vomiting bug
Mm-mm.
norovirus,
Yeah,
and
exactly. You
no
can like
way.
go on that cruise and not worry, you will not get norovirus.
Yeah. Eat all of the dodgy
Yeah,
food that you want, yeah, even if it's gone
exactly.
off for a few days. Yeah.
But then, on the flip side of that, because there's always trade-offs, non-secretors have a somewhat higher risk for certain autoimmune conditions like type 1 diabetes and IBD. And it's an interesting question to say, I wonder if that's related to the microbiome because,
Mm.
microbiome is starting to be related to everything. But when we come back to milk, sort of there are 25% of moms do not make 2FL, have a different microbiome or a different HMO profile in their milk. So what does that mean to their babies? Like, I think there's an inclination to say, oh, must not be good. They must be missing this HMO like for them. But I would kind of argue, well, they've been gestating for 9 months with this mom who's a non-secretor, eventually consuming these non-secretor HMOs through amniotic fluid. Like maybe they're just kind of primed to receive that non-secretor milk. And so two questions that emerged from that knowledge for me, one is around HMOs and their addition to formulas. As we talked about earlier, there's lots of additions being made to formula to make them closer to human milk. No surprise, one of these additions is HMOs in the last decade or so. Companies are catching on that these HMOs are really important. Let's start putting them in formula. But they're not putting 200 different HMOs, right? That would
Yeah.
be really complex and expensive. So they're sort of like, well, which are the most important ones? Oh, it must be 2FL because that's like the most abundant, which is true on average. But for 25% of people, they make none of this. So like, should babies of non-secretor mothers get formula with this 2FL that they would never otherwise receive?
Mhm.
Maybe, maybe not. The newer formulas are adding more than just 2FL. They're adding like 3 or 4 HMOs, which is, I think, a step in the right direction. But that's
And
something interesting to consider.
isn't the complexity of that in that we can't actually synthesize these are such complex chemical
Yeah.
structures that they're very, very hard to isolate or to synthesize, chemically.
Yeah, exactly. And so 2FL, in addition to being the most abundant, if you're a secretor, is also like kind of the simplest
The
structure
simplest
to
one.
make.
Yeah. Mm.
And so, yes, the more complex they get structurally, the more difficult it is to either chemically synthesize them, or there are approaches now, like could we engineer bacteria to create these things if we give them the right genes? So now that there's a motivation to create them for commercial purposes, there's a lot of work going on like how could we better make these like more efficiently and higher yields and also more different versions of them. So that's certainly in progress. I think it's interesting. The other question it raises for me is the scenario of donor milk. So for babies whose mothers are not able to breastfeed, or babies who cannot breastfeed, or be breastfed, like what do we feed them? For most part, the answer is formula. But for babies who are really fragile and vulnerable and born early and in the hospital, there's a recognition that they're at very high risk for gut diseases like necrotizing enterocolitis. And we know that giving them breast milk is the best way to prevent that. Ideally, their mother's own milk, but if that's not available, then rather than move straight to formula based on cow's milk, they will provide donor milk. So that's milk from another human mother who's got extra for whatever reason and decided to donate it. And it will get processed and pasteurized and then fed to those new babies. And so there's lots of fascinating research to be done in that area. But on the topic of HMOs, I sort of wonder, well, should we be matching the donor mothers to the recipient babies in the same way that we do for blood? Like if if you go donate blood, they don't mix that blood with other people's blood and then just give it to
Mm,
everyone, right? They say, what's
yeah.
your blood type? Let me give it to someone with a matching blood type. So
Yeah.
right now we don't do that for milk, but I wonder if we should. And there's, then the question is, well, how do you match it? There's all sorts of things you could match on.
Yeah.
But
So
for
it's
me,
not
the
just
biggest
secretor status.
question, yeah, but secretor status would be a place to start. So we actually have a
Yeah.
trial starting where we're going to do that and say, is it better for babies if we actually match their milk type on secretor status?
Cool. And can one, who is breastfeed or lactating, change the composition of their HMOs through diet, through, I don't know, probiotics or something environmentally? Is there any evidence to show that you can intentionally change it other than just these natural genetic
Yeah.
effects?
It's a tricky question. So we didn't find, for example, in our child study, much evidence at all of like diet, for example, being related to the HMOs. But in our study, we didn't have mothers, assigned to different diets. We just said, what do you normally eat? So at that level, there wasn't a signal, but there have been a few studies where they actually did say, okay, you mothers eat a high fat diet and you eat a high carb diet. And they do see some shifts in the HMOs.
Mm-hmm.
So it's certainly not as dramatic as something like, for example, the fatty acids in breast milk.
Mm-hmm.
So like omega-3 fatty acids are something that you can take as a supplement, a fish oil, or you can eat lots of fish. And if you do that and you are lactating, you will almost immediately see that reflected in your milk. You'll have higher levels of these fatty acids in your milk, and that will get passed on to your baby. So those lipids are much more diet-based or derived than something like HMOs.
Mm. Okay, so we don't kind of completely talk about HMOs because there's probably an
Yeah.
ingredient list about,
Oh,
200 or 300
yeah.
long yeah, exactly, that we could go on to. But I think the other one that's quite interesting, mainly because you'll be able to tell us more about this, because we can't yet replicate it in milk, is antibodies. We know that, I mean, you should tell us more, that the babies are protected from certain diseases because they're getting these antibodies
Mhm.
through milk. So maybe you can tell us about antibodies in breast milk a little bit.
Yeah, so there's a reason I'm cross-appointed to so many departments. I mentioned I'm pediatrics, but I have a affiliation to immunology. So I now work with immunologists, have immunology students doing projects on this type of thing, and then microbiology and nutrition. But so I'm more fascinated the more I learn about antibodies in milk. But essentially, when a baby is born, certainly if they're preterm, but even a normal, healthy, full-term baby, when they are born, have a very immature immune system. So they're not yet producing large levels of their own antibodies. That's something they have to develop over the first sort of year of life. And so that is also the period when they're drinking breast milk. And so they are getting antibodies from their mother. They're relying on breast milk to get those antibodies to help them fend off different infections. And the antibodies in their mother's milk is going to be a reflection of that mother's own sort of disease history and also her environment. So if she's exposed to a particular pathogen, and we could take the COVID pandemic as an example. So if mom's had COVID or someone that in the household has had it, her immune system is going to respond, create antibodies against that virus, and then the baby's going to receive them through milk. And in many cases, those antibodies are active in the baby and able to offer protection. And so, like you said, that's something that we can't replicate in milk. And even if we could, sort of, which ones would you put? It would depend, like, what environment this baby lives in.
Yeah,
And
because
the other thing
antibodies, I
is,
mean, for the antibodies are so much more diverse than we've talked about the diversity of
yeah,
HMOs, but antibodies are specific to individual, bacteria, individual kind of strains of bacteria. So it's almost
exactly.
impossible to replicate unless you put very generic
Yeah.
IgA or whatever's in there. Sorry.
And even those like, yeah, no, and that's where I was going to go next is with IgA. So the most abundant antibody of all the ones found in human milk is IgA, immunoglobulin A. It's very high levels in breast milk, and it's different than the IgA you would find in blood, even from the same mother. In the way that it's configured to be, we call secretory IgA. So it's sort of like designed to be ingested orally by a baby and survive that trip through the digestive tract and then be active in the gut. And it's doing lots of things beyond, protecting from infections necessarily. It's actually helping shape the microbiome. So this IgA can bind to bacteria, even like our good bacteria in our gut, but then help them kind of do different functions. And so this is something I'm working with Brett Finley and his team on, because a lot of the technology we have to study the microbiome is about sequencing the DNA of the microbes. So get the microbes out of your sample, whether it's stool or milk, and then sequence their DNA and say, oh, these are the microbes that are there. But actually, it matters a lot whether those microbes are bound to IgA, because that's going to allow them to do or not do certain functions, like bind to the epithelium or interact with other bacteria. So it's sort of a whole layer of information that's really important that has not been paid much attention to. And so the project I have with Brett is to look at, okay, let's not only sequence the bacteria, but also sort them into whether or not they have IgA bound, and then see what difference that makes to the gut microbiome community and the different health outcomes.
So does that mean, I mean, you spoke a little bit about antibodies against infections and pathogens. So if a mother, this is a way to protect the baby. If the mother becomes infected, she'll create the antibody, pass it through her breast milk so that the baby is protected. And it's a beautiful evolutionary mechanism. But as you've mentioned, antibodies also target the microbiome as well, the healthy microbes.
Mhm.
So does that mean that there are antibodies in breast milk that are specific towards the mother's microbiome, which then are passed to the baby and determine what types of microbes, normal microbes, colonize the baby? Is there an indirect kind of way of changing the microbiome through these antibodies?
Yeah, so this is an area that, still has a lot of work to be done. But yeah, that's the general idea, is that the mother's own complement of these microbiome targeted antibodies are going to, of course, be sort of reflecting her own microbiome. And if these are then passed on in the milk to her baby, it's going to be one way of her signaling her microbiome to the baby. The other way that we think of sort of more directly is that when a baby is born, it's exposed to its mother's microbes, both vaginal and fecal microbes. So it's sort of a direct transfer. We also can talk about the microbes that are present in breast milk. And of course, moms, kissing and cuddling their babies and having skin to skin time. So there's ways of microbes directly transferring from a mom to a baby. But then there's also this way of sort of the indirect communication through the IgA complement.
I think that's it. You've segued perfectly into what I was going to ask next, because we've talked about the sugars and these lovely antibodies which are there in breast milk, but a lot of your research is looking at the live microbes, the microbiome of breast milk itself. Um, so as you've mentioned, babies are exposed to microbiomes for the first time, microbes for the first time. I mean, you could argue, some people argue that this isn't the case, but really we know that the microbiome per se starts developing from birth. And a lot of people try to track where most of these microbes come from. And it seems like most of them come from mother's gut, some from vaginal microbes. But you have shown that a lot of these actually come from breast milk itself. Maybe you can tell us a little bit about what you found in the breast milk microbiome.
Yeah, so I'll first acknowledge that studying microbes in breast milk has an added layer of complexity compared to studying them in something like stool, just because of the sheer number of bacteria. So stool is like full of plenty of bacteria. Breast milk, as you would expect, isn't full of lots of bacteria. It has some, but they are relatively low amounts compared to, for example, the human cells you find in that milk from the mother's mammary gland just sloughing off. So if we want to use a method like DNA sequencing to understand them, we have the challenge of there's going to be a lot more human DNA in a breast milk sample than bacterial DNA. And that's not the case with something like stool. So that means we have to be very careful about things like contamination. Also, just because at such low levels, different reagents in the lab can be contaminated with bacterial DNA. So those are definitely issues to be wary of when you're studying this type of low biomass, we call it, sample. And we're very aware of that and doing the appropriate controls. So I'd say when I started doing this research, Shelley McGuire's lab in the US had published, I think, the first paper on the breast milk microbiome. And up until that point, it had been presumed that breast milk is sterile because it's coming directly from the mom, unless she has some sort of infection like mastitis, it's sterile. And Shelley's lab was the first to really analyze some samples and show, no, actually, there's microbes here. There's like live microbes. Her lab studies not only humans, but also cow milk. Actually her husband's lab. So they're a good partner, one doing human milk, one doing cow milk. And they had done some work to show, no, there are microbes in milk. They're there at a very low level, but they are there. And to show also that it's different between different moms. And so kind of following on the methods that she had applied, I did this in the child study in a larger cohort and also found like, yeah, we detect bacterial DNA. It does look like quite different between moms in a similar way that HMOs are different between moms. So are microbes. And so that we started asking similar questions about like, what is driving the difference in these microbiomes in the milk? And also, what does that mean for the baby? So first we were just describing and we thought, what's driving the differences? And we started with a list of things that we knew were important for the gut microbiome. So like C-sections, did it matter if a mom delivered by C-section or vaginally? And that wasn't really important to her milk microbiome. Maybe that's not a surprise, but we checked. We checked other things like the mother's age and her BMI. And none of these were like really lighting up the charts, but the one thing we found that was like the most important of anything was how they were feeding their babies. So they were all giving breast milk to their babies, but some of them were doing that directly at the breast, kind of all of the time, and some of them were giving pumped milk some of the time. And so that was an interesting clue to us about like, where do these microbes actually come from? And there's kind of two theories. One is they come from the mother's own gut microbiome and kind of somehow migrate their way into the mammary gland and into the milk. And the other idea is, well, actually, they kind of come from the outside, the environment on the mom's skin. And I think our finding lends credence to that hypothesis, because if the baby is always suckling at the breast, then the baby's mouth could actually be a source of bacteria. Whereas if the baby's never there and it's always a pump, then you get a different kind of exposure. And so that's also been confirmed by other groups now. So I think it gives, and I don't think, by the way, that it has to be one or the other of these pathways. I think both play a role. I think there are some internal microbes migrating, but also there are external microbes getting into the breast milk. So that was our first study, looking at what's there and what's affecting it. And then we wanted to know, like, how is this related to the baby's microbiome? So we worked with other colleagues in the child study who had already done the gut microbiome sequencing of the babies. And we compared the milk microbiome from the mom's milk to the gut microbiome from the baby's stool samples. And these were collected on the same day, which is nice. And then we said, to what degree do these overlap? And it's not surprising that they didn't overlap a whole lot, because the niche of breast milk versus stool or a gut is very different. So a lot of different microbes in these two niches. But we did find sort of a short list of about a dozen microbes that we found in both places. So they were in the milk and they were in the gut. And what's more than that is they seem to be correlated. So in a mother and her own baby, if the mom had more of microbe A, then the baby tended to have more of that same microbe. So we called that microbe sharing. And we were careful to say that because it's not clear whether the microbes came from the mom to the baby, or, like I just mentioned, the baby to mom. But regardless, there seems to be some sharing of those microbes going on. And so it's still an open question, like, what does that mean for the baby's health? But we do think those microbes are one way of moms sharing microbes with their babies.
And what are those ones that are most commonly shared? Because you've mentioned about these totally different niches. You'd expect like lactobacilli or something to grow in milk, whereas you get more like, enterococci or these firmicutes, kind of more things that are more, let's say, anaerobic, let's say, which want to live in stool. So what is passed most commonly?
Yeah, so you're going to test my memory now. Oh, maybe I'll open the paper.
You can get up your
But
list,
what I can say is, like,
roughly.
one,
Yeah.
yeah,
Yeah.
lactobacillus, you'd think, because it has that name lacto, like milk. But actually, it wasn't like one of the big ones we found in milk.
Right.
So maybe again a little bit of a misnomer. But we found, for example, and we were excited that on this like short list of a dozen microbes, two of them were familiar to us. And that was because we'd seen them come up before in the child study in research we did on the gut microbiome and asthma in children. And we saw that there were 4 microbes that seemed to be missing from the babies that went on to develop asthma, which made us think, well, maybe they're important in like setting up the immune system correctly in early life. And this was Stuart Turvey and Brett Finley's group, and they nicknamed those 4 bacteria flavor, FLVR for the first initials. And so the V and the R are Veillonella and Rothia. And those were 2 of our bacteria that we found shared between milk and gut. And we knew from other research, still in the child cohort and others, that breastfeeding seems protective against asthma. And the question is, like, how does that work? Is it through the microbiome, or it could be other things? And we've always thought the microbiome was a likely pathway, because we see, breastfeeding related to the microbiome related to asthma. Is this like a causal pathway? So this provides some evidence for that. So bifidobacteria is another, and that's one that we hear a lot about in infant microbiome research. We know that breastfed babies have microbiomes with high levels of bifidobacteria, depending where you look, even highly predominant bifidobacteria. So that was one that we did find in the milk as well. Staph and strep are commonly found in the milk, and maybe they come from skin, but we find those also in the baby's guts. So yeah, kind of an interesting list. And for me, I thought too, it hasn't been my field of research to like develop products, but if I was looking for like, what bacteria should we use as probiotics, like ingestible probiotics, I would think milk would be a good inspiration for that, because just in the same way we talked about the IgA antibodies in milk are sort of configured in a way that's designed to withstand digestion. So might these particular microbes, because just because you eat a microbe doesn't mean it's gonna like survive the trip and set up shop in your gut, but maybe these ones do. Whether that would work for adults is another question, because baby guts are very different than adults. But
Yeah, I can picture, it's probably like the gut microbiome, when people ask, what is a healthy microbiome, or is my microbiome right? It's probably similar in milk. I'm imagining that we don't
Yeah.
know for sure what is healthy or not yet. We know there's different microbes there. So, for example, if we take your pumping example, you found different microbes in the milk from women who pumped versus those who breastfed. I mean, what could you say that was a worse microbiome, or could you say, what were the species there that maybe would allow you to say whether how bad it was, or whether it was bad?
Yeah. So you said, could you say that it's worse? So I wouldn't say that it's worse, but the media would say that. And I learned that lesson the hard way, right? We put out this paper and then the headlines were things like moms who pump have worse bacteria or are causing asthma in their babies, which, is not what we said, but things can get out of hand in the media. So I'm always careful when I'm giving a talk on this topic to like take a pause and a PSA slide and say, I'm not saying pumping is bad. I know, secondhand from friends who pumped, it's a lot of work. And our own data show that, babies who receive pump milk are still receiving benefits from that milk as compared to being fully formula fed, for example. We see differences, and I think that's important to study and to understand, is it that pumped milk, you got to put it in the fridge? Are some of the microbes or other bioactive elements losing their activity? And once we understand that, could we give better instructions for storage? Or is it about the lack of that skin-to-skin contact you get through breastfeeding? And if that's the case, maybe even if you have to pump, you could just make sure you have lots of skin-to-skin time with your baby. So there's lots to learn there, and I think it's important. And we know that for some moms, whether it's because they need to be at work or their babies don't latch, like pumped milk is what their babies are going to get. And so with that out of the way, I will say we did find some higher levels of potential pathogens in the milk from moms who pumped. And so we speculate, that could be an issue, which again is an actionable advice of not cleaning the pump and apparatus appropriately and having those microbes feed back into the milk. But the other thing is we call them potential pathogens. Like they are bugs that have the potential to be pathogenic, but that doesn't mean they always are, right? Some babies can carry this and not get sick. But yeah, that's something we're looking at. I had a former postdoc, Sarah Reyes, in the lab who had done her PhD specifically focused on pumps and pump hygiene and how pumps were cleaned and what that meant to the microbiome of babies and moms. So I think it's a really important area, especially in certain countries with little or no paid maternity leave, where that's the only option.
And so is there any evidence, you spoke about this fascinating potential for microbes from a mum's gut to go through what they call this enteromammary axis and travel through the body, potentially up into the mammary gland. As far as I know, there's no evidence for this in humans. Maybe there's a little bit of evidence in mice. But does that suggest potentially that if mothers take oral probiotics, some sort of strain, which probably doesn't exist right now, that could change the microbiome of milk if they wanted to do that for whatever reason?
Yeah, I think like you said, there's not much evidence for this yet, but certainly the possibility is there. I think, yeah, there's been a bit of evidence in like mice or maybe cows tracking orally administered bacteria, finding it in the milk. And I think there's been some work in mice with sort of fancy fluorescent tagging and tracking of like immune cells from the gut and seeing them actually physically go to the mammary gland, which I think is fascinating. So to find out if this is the case in humans, I think is an important question and really interesting. But yeah, I think there is the possibility of that. It would be, like you say, a matter of also finding the right microbes that are able to survive this different translocation and doing more research first to understand like how is it that certain microbes get selected out of the mother's like complex microbiome to then get translocated to the mammary gland.
Mm.
So a really neat
Or is
area
it a
of
case,
research.
I mean, yeah, with all the caveats you're saying that we don't know what is a good breast milk microbiome or not at the moment, you're still at a stage in your research of kind of just identifying these differences first. But, looking a few years down the line, maybe from this reach, you'll find something that suggests that maybe something is missing in certain women and maybe there will be something that needs to be replaced. So that could be orally. Equally, it could be some sort of like topical skin probiotic. You said that some microbes, they come, you think, from the environment. You don't exactly know where, maybe from the skin. Is there any evidence for that, for like, showing that skin microbes, topically applied, if they're probiotics, can
Yeah.
have beneficial effects or make their way into breast milk? Or
So that is a great question. I'm not aware of any work going on in that area, but yeah, like for example, there are antibiotic nipple creams for if you have a mastitis infection. So I could see a world where there's probiotic nipple creams that are gonna, help help the breast milk microbiome. Like it's possible. Also, we could think about if the goal is to get this ideal healthy microbiome formulated in the mom and then in her milk and then to the baby, like maybe you skip part of that step and say, let's try and give some microbes right to the baby. And certainly there are people working on that, probiotics for babies. But I think one of the things to keep in mind then is like, well, the way a baby naturally ingests microbes in breast milk is that many of those microbes are going to be IgA bound, for example, like we talked about earlier. So if you just give them the like naked microbes, maybe that won't have the same effect. So maybe we have to think about, or also too, like if this baby is not being breastfed, then they're not getting HMOs. So do we need like IgA bound microbes with HMO, like what would naturally be in milk, which is so complex and we're still learning about. And that's only 3 components we're talking about, right? Like there's also certain lipids and other metabolites and short chain fatty acids. Like these are all found in breast milk. We're still kind of getting the full ingredient list.
Yeah, yeah. So what is the other exciting thing? What have we not touched on? You've mentioned a few
Mm,
there, which, what's next going to be added to the list of formulas to be replaced? It's not going to be antibodies. It might be another
yeah.
HMO, but I mean, is it a lipid? Is it a metabolite? What's there and is very
Yes.
important.
Well, I'll answer that in 2 ways. One is that I always like to say, rather than like, what's the other thing we need to add into formula? I always like to say, like, let's dedicate some energy to promoting breastfeeding and doing what we can to support moms who want to do that, but maybe don't have the support they need. Because, I have a quote from a colleague that I always love who was asked, like, if we put this in breast milk, will it be like better, closer to humans? And he goes like, yes, in the same way that if I stand on my roof, I will be closer to the moon. Like I will be closer, but I'm really never going to get there with that strategy. So I think we need to keep that in mind. But at the same time, I think we do need to study and learn more about human milk for the reason of making better alternatives for babies who need it. Also for many other reasons. Like there's a lot we can learn from milk. Good example, since we talked about antibodies and COVID, like there are ideas of, well, could we from donor milk, like extract the antibodies and then use them for treating like COVID patients or preventing COVID in elderly people who are at high risk. And COVID's a little bit behind us now, but for the next pandemic or for other diseases, or it's been found that we think we talked about earlier, milk is supporting immune system development for babies who have a very naive immune system. I think like who else has a very naive immune system? Well, people who get bone marrow transplants, they first have to like wipe out their immune system basically so that they won't reject the new donor marrow. But that brings with a lot of problems when you wipe out your immune system. So there are studies going on, like, could we feed breast milk to those people to support their immune system in a time of need? So for all these reasons, I think there are lots of reasons to study what's in breast milk. It has a lot to teach us. And so one of the projects I'm really excited about is I have a project right now called the International Milk Composition Consortium, or IMIC, and we're studying milk from women in Canada, but also Tanzania, Burkina Faso, and Pakistan. So very, very different environments to try and look at what's similar and what's different in these different contexts. We're measuring growth outcomes in the babies. So 3 of those places are quite different to Canada, and they're environments where stunting and wasting and like poor growth is a problem for babies. So understanding if we can identify profiles of milk related to optimal growth, and then that can teach us how to support other babies. I have projects on food allergy prevention and donor milk I mentioned. Another one's antenatal milk. So the idea that moms can actually start producing milk towards the end of pregnancy. And right now that's recommended in certain scenarios, but it's also gaining a lot of traction, which in a way is exciting to me. Like people are figuring out that breast milk's important. If we can get some of it like stockpiled before the baby, like maybe that's good. But I also think a little bit like, whoa, no one's really analyzed that milk. And I don't know like what the composition of it is. So I want to look into that. Like there's just never ending projects around milk.
It sounds like you've a long career yet to go anyway before, and
Mhm.
surely none of it will all get answered. But it sounds like you're doing some very exciting stuff, and you've given us an amazing insight into breast milk. So I think we'll leave it there. And thank you very, very much, Megan, for giving us that insight. And we'll surely hear some more in the future from your ongoing research. So thank you.
Awesome. Thank you for the work you're doing to sort of spread the word about microbiome science. I think that's a great service to the community.
Great.